Hello, everybody. Welcome to the Roach Wiggle, December 8th, 2022. I just realized as I sat down here to start things that I had not made the thing. I did not start the live stream or get it set up for this week after last week. So I'm actually going to go ahead and schedule for next week, the 15th, and just get that slotted in here real quick so that I don't forget. And I'm assuming I do have to check, which I will be doing this Saturday. Also, this will be a very short live stream, perhaps even among the shortest. I will explain why very shortly. A lot of short things. But this weekend I have an expo, and thus that is part of the reason. And I'm going to be asking Kim if not next week, but the following week, Thursday, the 22nd, will work for him for a guest live stream. And then at some point, Kai and I do need to sex some centipedes again and do something like that. So that's going to be that's on the docket as well. Just getting all this here. YouTube seems to have changed a couple of things in their stream creating interface. Not too much, not throwing me off too much, but just I had gotten cozy with not screwing things up with the old layout. So okay, that's scheduled. So we're all good there. Let's go ahead. Let me go ahead and say hello. Hello, Chain, Jojo, Tennyson, Jose. I enjoyed the wiggle. The wiggle acronym is great. That's wonderful. So let's see, where are we this week with this incredibly short live stream? So I wouldn't be surprised if I limited it to a half an hour. We'll see what happens as I start talking. It has been a crazy week. I have been, Javier has not been able to come in, so I haven't had the extra muscle for some things. I have, however, been very, very, very task-oriented and diligent, and I have finally gotten to working on my millipedes. Those who have been listening for the last couple of months have probably heard me saying, oh yeah, I still need to do my millipedes. Oh yeah, I haven't done the millipedes. Oh, I haven't done the millipedes. And I haven't really done any decent work on my millipedes since like maybe right before the Arizona trip. So they really needed that. And so there's a couple of things. Redemption. Junior, speaking of millipedes, I have some news for you. You know how I said I didn't have any purple ghost nasodillo? Well, what happened was at some point, and I just have so many containers, even though everything's labeled, I still don't know 100% of like the ice spot stuff. I don't have a master list. I will be working on that. Anyways, where I'm going with this is, I apparently at some point pulled them all from the millipede enclosure that they were living in and set them up in their own culture. So then I was going through and pulling some stuff for the expo this weekend, and I came across the containers like, what the hell? Like, I didn't know I still had these, and they were doing quite well. So in the future, if you do decide you want to get purple ghosts, I do have them. Although honestly, the more that I look at them, they're just you will be much happier with both of the species that I sent you, and instead they're just so much. The colors are just so much cooler. Let's see what Tennyson says here. That reminds me, I have seen some people keep hissers and millipedes together. Are there any specific species that would do well with port and toes? They have enclosure moist, half bone, dry, about 78 degrees. Just about anything if you make the substrate deep enough and you have enough space. I keep my hissers so like warm and cramped and everything that I have isopods with almost all of my hissers. But for my purposes, it's not a good combination those two because of the way that I keep the things for the hissers. Because I love my hissers, and thus they must get the best at all times if possible. Splendid, yeah, Centropolis Splendidus could work because they do seem to be just hardier all around. But you got to really make sure you accommodate for the millipedes. I'm sure the hissers would probably avoid the cooler, moister end naturally. So, Junior says, "I'll let you know." Loving the white and blue pigeons though for sure. Yeah, the white pigeons really grew on me once I got them. Kai got them from someone, and I was like, "Kai, give me these. You're gonna kill them." And even though he was actually doing quite well with them, and then I got them really going, and I finally got to see them full size. Like, yeah, this is pretty neat. They have it's like an ivory coloration and a little bit of faint striping. It's pretty neat. It's better than a lot of the other ones. I got some species soil from Kai, and I just think the adults are gross, but the younger ones have little whitish antennae, and I thought that's pretty cute. It's kind of like the Porcelio species Almeria Mountains. Yeah, so let's see. So I'm currently haven't had to do a big crazy isopod revamp of things. These new setups I really feel like have been the way to go. Very satisfied with the results that I'm getting in pretty much all the species that I'm keeping in them. So I've just been bulking out the numbers of stuff that I have. There's what is Corey's Crazy Isopods? He's a pretty neat guy who goes to the same expo that I do. Every month we usually end up swapping for something. Like there'll be something that I've killed or neglected, and he'll have a little bit of it, and I'll get it from him. And he's got pretty good so far. I haven't found anything wonky with the stock that I've gotten from him for different simple recessive mutations and stuff. So it's neat. He's a neat guy. If anybody's looking for some isopods I don't have available, maybe check him out. I think that's the name of his business. But anyways, this expo, I'm really going over the top because it's Christmas time. I haven't really gone over the top for an expo. I feel like I have an opportunity to do that just because of I've just a lot of stuff. Next week, I don't have much shipping stuff going on except for Chris's order, which is the last auction order going out. And by the time this is all said and done, it's going to be March or April, and I'm going to be ready to do the next auction. Yeah, so it's something. Yeah, so just bulking out, splitting some things. I've rearranging the base. This is the whole thing that I'm in, and why I want to do a shorter live stream this week, and who knows about next week. But I'll probably have a better handle on things by next week, because I'll have had Monday, Tuesday, Wednesday, Thursday to keep working on things. Javier might come over next Thursday too, but basically, partly because of the OCD voices in my head, but also just for logistics purposes, I've had one rack of millipedes, and it's kind of backing out of the way in the basement, and it's also along the wall that is slowly being monopolized by isopods. And so I've decided, they're not getting enough attention there. Obviously, it's a pain for me to go and actually take care of them in that location because they're heavy tanks and they're out of the way. And so I need to move them to a different wall. So I've decided that while I'm in the middle of doing all this, redoing the enclosures or refreshing all the material and whatever, that I'm going to take them all, work on the containers, move them to a temporary spot, and it's awful. I have 5.5 gallons crisscross stacked on top of each other. It's horrible. It's horrible, horrible, horrible. It's just in the most inconvenient locations, but it's just I just got to get it done. Just got to get it done, and it's going to be done. It's going to be done. And this will allow me to have that whole wall be the isopod wall. And I think each individual shelf. Javier worked in food processing or food what was it not, I'm trying to think what you call people who work in not chef, but you're involved in hospitality. There's a specific word for like people who are like helping organize and run kitchens and stuff like that. Anyways, but he's been calling them metros, which is easier for me, I think, if I remember to use it, because that's a brand of shelf. But I always sometimes get rack and shelf mixed up. Maybe serve restaurant service industry. Hello, Keaton. So each metro, each shelf, I guess I'll just say shelf. Each shelf holds about, I've probably gone over this before too, 60 to 100 containers. I mean, you can kind of see it here, but there's more on this one because of the smaller size of these. But so yeah, the whole wall is just going to be infested with isopods, and I'm getting to the point where I really do need that extra rack for more, because we just cleared off the end cap rack on the one side, and it's already almost filled up. So now it's like the second I got more critter keepers in, I was going to start working on the other end, and I just got a whole other shipment of them in today, with like the critter keepers falling out of the box when they threw it on the front porch, because it's just a gigantic box full of critter keepers. So that the gears are turning on that. So it's just all these things that I have to orchestrate at the same time. It's very stressful for me because I have to tell myself, don't clean up, don't rearrange, don't do this stuff. You're in like the middle of doing things that, you can take care of all these little details and stuff later after you get all this other stuff done. So all the isopods for the expo are packed up, and the roaches are what I'm working on now. And I'm also simultaneously doing roach husbandry, my monthly roach husbandry at the same time. And so it's a lot of things. Tennyson says, do Venezuela parvis and Armadillidium pulchellum breed at a similar rate? No, the pulchellum are much slower in my experience, and maybe more picky about, like, Venezuela parvis will colonize coconut fiber, and with the proper conditions, you will just end up with a substrate that's all Venezuela parvis. But I have not seen that out of the pulchellum yet. And I've had them for maybe a year and a half, 2 years. So yeah, and apparently they're not very common. A lot of people lost them after they were first brought in. So they may just not be a very aggressive fecund species per se. So isopod stuff, the millipede stuff, and the cherry on top of all this is, and TJ may show up later, he may show up after. Hello, Brett. He may show up after I end up ending the stream. But TJ sent me a lovely box of goodies today. I feel very strongly about what I was about to say, and I almost broke my code and heavy swore there. I am extremely tired of killing or not doing well with Plectoptera poii, and so I told TJ, I want all that you can send me. Just load up a container with it. I don't care if it's hundreds or thousands. Just send me all of them that you can. And so he sent me a box with a bunch of really good goodies. Parker Americana from Table Rock Mountain in Idaho. Really fun new things, like these blattids from TJ. If you're watching this, I can't remember where they're from yet. I haven't fully processed it. They might be a Doralea species, actually, which is really exciting. We don't often get new blattids in culture. You think blattids is like Paraplaneta, Shelfordella, Blatta, Derapeltis, and then Doralea orani, and I think that's about it. That's all genre that we really have. Uricotis, I guess, and all those associates too. But those don't even really feel like they're not the Blattidius. Also, speaking of that, everybody's been letting me say this for months now, but I found on iNaturalist they have Uricotis listed in their own subfamily. Yeah, so on the cockroach species file, they recognize Uricotis as belonging to Polyzostriinae, and iNaturalist is listing them as their own subfamily, Uricotinae, which I thought was weird. or I mean, obviously there's the new world, old world split there, which you can make an argument for a lot of things with that. So, but the species file hasn't updated, so I don't know. I don't really know what to go with. So I will continue to use Polyzosterini until I am corrected or until species file updates. And Junior, yes, it might be the species Philippines. Brett B says, I accidentally obliterated my first group of Poya, but now they're doing good after buying 30. Yeah, a lot of little ectobids will be like that. So easy to kill them if you don't have enough of them or don't really have a full understanding of how to maintain them. And then like now I'm at the point with some stuff with like Neoblatella detersa where I don't even want to open the container. There's just so many of them that it's a stressful event. I do it as little as possible. So Junior, I think it's that. I think it's the species Philippines. So I've got some of those, a couple of other things, and hello Hylas, good to see you. Good to see a lot of people in here. Again, this will be a shorter live stream. I hate to say that, because the expo this weekend, and I'm in the middle of killing myself by doing a bunch of things at the same time that probably should not be done simultaneously, like moving millipedes to a new section of the basement and taking care of roaches while packing up stuff for the show at the same time. So anyways, updates on sabbatical. I can hit that really quick. Have not had any donations really since I had last done the updating. Obviously, I'm not trying to beg or ask, but I feel like I'm coming to the point where I gotta put my foot down and either say if I'm going to take that week off or not. And I'm thinking that I'll make that decision by the late Sunday or Monday, and go from there. Really, it's overdue. It really needs to be done. And so, regardless of whether or not I hit the goal, I think I should probably do it. I mean, I've talked it up enough, and people have donated, and I don't want to let those people down. And it's a good time of year to do it too, with the risk of shipping that close to the Christmas and whatnot. So it will most likely end up happening. So everybody can look forward to that, and some ramblings and some updates on some things. Looking around the room, anything? I think since last live stream, I did a check on things in here. I think a lot of things are doing well. Lots of Hemithyrsospora vittata. I might actually do a giveaway for those next week or the following. One of these weeks, I'll probably do a giveaway in the stream for something. I have to. It's Christmas, so I have to do some sort of giveaway. So everything else. This is pretty good news, actually. Euthlastoblata abortiva. I found babies, which was this has been a very coveted U.S. species with, I think, a restricted range in like southern western Texas, and very little information on exactly how and where to find them. It's like there's like a half-eaten or half-broken specimen on Bug Guide and a record that like, nymphs have been found in like wood piles and rodent burrows or something like that. It's not really much to go off of for trying to find something, but I think I brought this up before. I received a bunch of adults from a contact. I'm unsure if he wants to be mentioned or not, but from a contact who isn't really keyed in with a lot of the main roach people, but he is a very, very cool entomologist, keeps a lot of parkablata and things like that. He happened to be collecting with permits from a state park in South Texas and found a bunch of them, sent some to me, was absolutely elated. They look like teeny tiny Paraplinidae australasiae or Dorlea orani. And they're really interesting. Seem to be easy so far. Don't seem to have any sort of gimmicks, etc. Catch up on comments here. Chain says, just add some input. I couldn't this week, but it will be up by 10%. Oh, thank you, Chain. I appreciate that. Again, I think that if I left it up long enough, we would hit it, and that's why I feel like, I need to just commit to that week and do it regardless. So I have no intentions of giving away my armpit hair, Keaton, although I could see that being something that would happen at like the Tinley auction. somebody would auction something weird off like that. Wouldn't be the weirdest thing that's been auctioned. Brett says, it's an interesting looking little guy. Yeah, I really like them. They're very different from a lot of the other Euthalastoblata, and again, seem to be pretty easy. And that's enough of a selling point for me. And just the fact that I think after we have, if those get established, then I think the ectobiids that are really left that are kind of like crown jewels are like Plectoptera picta, Latiblatella leucophrons, which I figure I'll just stumble across again in Arizona one day. It'll just happen randomly, probably during a very moist year. And that's about it for like ectobiids. I mean, there's a bunch. There's like introduced Compsotes and it would be really nice to find the leafcutter ant cockroach. That would probably need some very intricate timing to be there during a nuptial flight, or to find a group in higher education that's going to be dissecting a leafcutter ant nest for some reason and be able to get into those fungus chambers and just grab them out of there. So maybe one day, maybe I'll get enough funding for something in the distant future. I'll be able to take an extended trip to Arizona to try and collect them and culture them, and probably end up failing with them once, then have to do it again, find them a little bit easier, and then hopefully get them into culture eventually. But and yes, Brett, I do really want Liridoblata trivittata. They're kind of hard to get. They're just kind of hard to find somebody who finds them reliably and will ship them. I have had them before. They're pretty easy. They're like they don't seem to require a diapause. But the ooths do seem to, similar to Ectobius pallidus, prefer or need a wet or moist dry cycle where the eggs have to desiccate and then take in water and then they'll hatch. So I have hatched them before. Cody Will from Cody's Bug Room? I can't remember. It's been a while. Or the Roach Lab, maybe? He was out of Arizona. He originally sent them to me a while ago. I think it was 2016 or 2017, and I was able to get babies out of them, but obviously didn't put in enough effort to sort of keep them going, because it's not really that bad. I have a good colony of Ectobius pallidus going now, and so I don't feel too intimidated by that. But it's one of those things, you gotta give them enough space in a container to like osmoregulate, like these containers here, these sort of styles where you can keep it one side wet, one side dry, and good ventilation. So if I get them again, I'm sure that I will get a good colony of them going, because some of that natural like dry moist cycling just naturally happens with like my spraying schedule sometimes. So I'm not concerned. It's just a matter of acquiring them. So everybody here, I'm going to look up Liridoblata's range. Liridoblata trivittata. Wasn't it recently changed to a different genus? Maybe, maybe not. Called charismatic cockroaches. That's cute. In this article. Friendly cockroach. Yeah, so they're in California mainly. I think I'd heard of them in Nevada. So yeah, anybody knows anybody who can collect Liridoblata? Very interested. They are very cute. There's some very adorable pictures of them. I mean, honestly, they seem like they probably fill a similar niche to Loboptera. Oh, there's a record from Texas. That's probably not actually it, though. Needs ID. Yeah, it looks like a Blatella. Although it's a very crappy picture. It's probably a Blatella. So they're concentrated around San Francisco, north to about Redding, east to El Dorado National Forest, and south to, there's a lone data point in Bakersfield. Let's see if it is a Liridoblata. Could be, couldn't be. I don't know. Definitely not the best picture. This is almost certainly one, though. Yeah. So anybody who's in that area of California has any connections, Loretta Blada, would love to work with them again. Should be pretty easy. Chain asks, do the little penguin roaches have a problem with crowding? They keep eating their ooths, and that's the last thing I haven't tried yet, giving them more space. Let's see if I can illustrate a point. This, let's see, I got to get it close enough. And then should refocus shortly. Should, maybe, about right there. Right there. Right there. This colony, I restarted with stock in March, and there's like hundreds in here now, and pretty much on top of each other. So I don't have problems. I don't think this is a species that has problems with crowding. You can pretty thorough infestation of this container, if you ask me. So it's probably could be diet related. Oh my, they've eaten. I put like a gigantic piece of apple in here, and they've completely eviscerated it. So all right, it's jelly time. So it could be diet. It could be the state of the stock when you received it. This is something I'm going to go into in the care guide too, because it's very important, and it's something that is not really, the importance of which has not really been emphasized, and it explains a lot of things that can happen with roach keeping and cultures. And I think I want to, I'd like to get some jargon terms going for roaches. And we have a couple, like, we have like ooth and whatnot, which are derived from, pre-existing terms. We have, and bladder composting, which is what I came up with to describe using cockroaches for composting. But I feel like we need a couple more jargon terms or things like that. And one of those is that I'd like to use is, where's the boy? Is death spiral. I would like to start using death spiral as a technical term to describe the phenomenon of roaches that are either that suddenly the colony suddenly begins to collapse, and it's usually due to cumulative effects of overcrowding, of lack of food, of improper conditions, and you can very easily lose a gigantic thriving colony and then proceed to not get them to pull out of it ever, or only with several years of time get the colony to recover. And it's a phenomenon that I've seen. Not genetic defects. These are cumulative effects that are not genetic, but may be heritable in some capacity, such as nutritional deficiencies, pathogen loads, or sort of elite effect related things where or imbalances of the gut microflora, the gut microbiome, which is huge with roaches. There is, I think it was Alex Baranowski had linked me to something where, and I brought this up before, where take something like lobster roaches, where you can have a container of like thousands, hundreds of thousands of them. They do excellent for just long periods of time like that. And they can be on top of each other, and you can throw anything in there. You can throw literal garbage and they'll eat it, and the colony will keep reproducing and be happy. And then you may change something knowingly or unknowingly, like say you take a pair out of that container and you put it into another container the same size with the same conditions, and all of a sudden they won't produce babies, they won't do anything, they won't eat, they die early. And you can have this sort of happen in multiple different ways over time. And what can happen is, no matter what you do, and this was studied specifically in lobster roaches, where they tested to see if you could rescue individuals that had poor nymph conditions, and if they would mature and be able to reproduce and be healthy. And basically was at some point there's so much damage done in some way that it's nearly impossible to get them back on track. And so I think I'm gonna figure out and coin some good terms to describe different parts of this process, because I feel like it's probably what happened with things like Langstroblata and Junior. I got them from you. This is no offense to you. Don't take this. This is not personally, because I have killed countless cultures of things and have had this happen countless times. I'm simply using it as a point to illustrate what may have happened. But I got a group of Langstroblata and was taking care of them fairly well and got them, a lot of them mature. And at some point even got them to produce a couple of offspring. But no matter what they were fed, no matter how they were kept, no matter what was changed, could not get them to just thrive. There's just this something that had begun to happen, which would explain something, whatever happened to your colony originally, whatever the original trigger was, Junior, that that just could not be overcome, even if you fixed your husbandry, even I fixed my husbandry, etc. I've had this happen with some other things before, Paraplaneta brunea, but even Botella germanica, have had that happen with those two. And so it's a very interesting phenomenon, and it would be very valuable to figure out if there is a way to overcome it. Whether it's like supplementing with electrolytes or something like that. Mark Mayer, who hasn't been on the chat, and he's not very active with a lot of bug people, but he's made some breakthroughs with some roaches and some miscellaneous invertebrates. But he'll sort of put two and two together on some things, and was telling me about how he had success with getting the condition of pill millipedes to improve by offering some sort of electrolyte and probiotic mixture. And yet that's another thing, is where you can just have a bunch of females that look perfectly healthy and happy and great, and you just will not be able to get them to reproduce. And then sometimes you look out, you get a couple of them, and then, once those get to size, they seem to have recovered from this, or once they get to reproductive age, they may recover from it, and then the momentum keeps up. So yeah, and it's something that I feel needs to be talked about, because some people will be discouraged if they fail with something, and they like, oh, I did everything I could, I followed the guide, I took everybody's advice, I set them up exactly like I did when I had them 7 years ago, and they were really thriving. And it may end up being that it was kind of out of your power at some point. So I had that. Here's another example. I had that happen with the little penguins. My husbandry for them was on and off for a period of time, and either they got too moist at some point, they got too dry, and it screwed them up, and I was down to just a couple of females. I set them up in this very same setup that I have over here, and expecting the best of results. And just nothing ever happened. They just never reproduced. The ooths that they produced just were not healthy, and they never hatched any offspring. There was a male that I added at some point. The females ended up being infertile. Just nothing that could be done to pull them out of that. And then starting over with, I don't know, it was like 10 to 20 or something of them. Threw them in the container. No problems. Not a problem in the world. Not a problem in the world since I set them up, as you all can see. So... and it can happen from everything from little ectobids like that all the way up to Blabberus. I think that may have been what happened with my CF chacoensis colony, was at some point either my husbandry schedule or something like that, or maybe the food that I was giving them or the environmental conditions, they entered a state where they could not be salvaged, and then the colony just died off from there. So, and sometimes you got to learn what can be done to prevent that. Sometimes it's as simple as making sure that there's always food. Sometimes it's not giving them too much water. Sometimes it's not giving them too little water. The devil's in the details sometimes. But there's the point that sometimes things can get screwed up so bad that you cannot pull them out of the spiral that they've entered, or you can, but it's going to take a long time to get them somewhere. So, So yeah, that's fun, fun topic to discuss. Inevitable failure, yeah, it happens. You just gotta, move on with it and hope that one of your bug buddies has a colony going still, so you can get a couple to restart using that. What else did TJ say? Episymplos syndaica, which is another cockroach that will probably join the ranks of roaches that I will actually never have a sale for because it's just nobody likes Simplos and Curiosa, and Episymplos syndaica is like pretty much the same thing visually, and I can't even remember if the females are fully winged or not, but it's another small orange ectobia that doesn't necessarily have any sort of standout qualities to it at all. So, hello, Chris. A Paraplaneta missionary has joined us as well. How are the venom tons of them. Tons of venom. They're doing amazing. I'm actually itching maybe to do a cross of Paraplaneta Americana Black to just a wild type strain. Some people who've crossed those have said they produce some brownish individuals, and I'm curious if something could be done with that. Currently working on making multiple Blatella germanica lines of different colors, and that seems to chocolate. Blatella germanica chocolate is good to go. If anybody wants some chocolate colored German roaches, they're good to go. I think I did the last selection maybe like right before Arizona or something like that, and a bunch of inbreeding will do that. You'll get a nice stabilization of phenotype sometimes, and that's what's happened. So if anybody wants chocolate colored Germans, they're here. They're ready. I got to add them to the site eventually, but they're good to go. I am working on recreating the original black German, or as I think Alan has called them, Hitler's nightmare. Recreating the black German strain. So the original one died out. I actually used males from that original strain to create the chocolate, which was for some reason crossing them to those, where are they from? The Alexandria, Virginia Blatella germanica, apparently. Hello, Braden. Apparently, whatever the dark gene or whatever the genetics behind the solid black Germans, just I could not get it to stabilize in these when crossed with these Alexandrias. So the chocolate Germans are descended from the original black German line, but I could not get that really black dark phenotype out of this cross. And so instead I decided to double down on selecting for these chocolate colored ones, and that's worked out very nicely. Hello, Kai, and your whatever you are. But anyway, but I have the stock from which I originally refined the really nice black Germans from. The really nice solid black ones, which are the ones that are pictured on the site, where all life stages are solid black. The adults are black. There's no traces of other coloration. And so I do have the stock that descended from, and it has produced some very nice dark individuals. So I pulled one of those to begin the selection process. Let's be hopeful, and maybe by June I will have some nice solid black ones available. That would be nice to have a couple of. I'm also working on a line I'm calling Amber. I'm not sure if the color is the result of genetics or if it's due to a Serratia infection, or if it's a complex interaction where I'm gradually making the line more susceptible to Serratia infections, and thus the phenotype is looking lighter and lighter. I don't know. We will find out eventually. Paraplaneta missionary, if you want a black and white Paraplaneta, you just look at Doralea orani, because the nymphs are basically like black and white, and they're sister genus to Paraplaneta apparently. So exciting German projects. Really wish that I would end up with something. I wish I could get like the white-eyed Germans, even though we have white-eyed Blatella vega now. White-eyed Germans would be cool again. The ones with the hemolymph bubbles, CPI. There's all kinds of mutations that can pop up, and you just got to get lucky and hope that you can find it and pull it out, because the lab or person who was originally maintaining all those really cool fancy German mutations, which you can stumble across in Google Images, Apparently they retired or they stopped working in that lab, and nobody maintained their cultures, so all of those are extinct. You can't acquire them, to my understanding, unless there's any private entities or universities that purchase them and have been maintaining colonies. Which, let's be honest, if a university is going to maintain a colony of a cockroach long term, it's probably going to be German cockroaches. So yeah, Brett, they had heritable hemolymph bubbles, which is both gross and cool. I have seen there's a mutation. I believe that it was genetic. I don't know if all presentations of that phenotype are genetic or if some of them are environmental, but it's where the wings scoop up at the back. So it kind of looks like a tuxedo or whatever, like a pianist's outfit where they have to like flip the tails up in the back. So I have seen that pop up in a couple of Germans. I haven't really gotten the desire to pull it to work and see if it's genetic or not at this point. Maybe I will lose more of my sanity and decide to do that, but not now with all the stuff that I have to do. So, yeah, so it's, again, busy week. Expo this Saturday. Next week is going to be just hit the ground running. A lot of boxes to check, and then I will have my sabbatical thing the following week, which I guess I should declare that on the site pretty soon, where I will be ignoring the world for the week, except for the live stream, and working on the care guide and getting it up hopefully by the end of that week, or sending it to everybody who donated first to sort of peer review and ask for some additions and sections, and then hopefully before Christmas get that out and about. We also have next week's live stream. I'll post something in here so that people who are watching the dead stream can also can key in on this. Next week, I would like for requests for stuff to see for a bug room tour. So I'm thinking I'm going to do. I had brought this up before. Nobody seems to have had strong opinions on it. I was thinking about doing a live tour, but then I also realized that the video quality will be poorer, and I think a lot of people would really be looking forward to this. So it's probably better for me to pre-record it with a sort of list of things that people wanted to see, and probably do it as like a special upload, at the stroke of midnight or something on New Year's. So I would like anybody who wants to see anything, if you want to see my Paraplaneta collection or anything like that, next live stream I will start taking notes on what people want to see, and then I will go through and record it when I do the tour recording. Of course, I have my own idea of what I'd like to show, but I think people want this, and this will be a better way that people can get what they want to see, and it'll be a nice HD quality and whatnot. So let me go ahead and write that here. All right, so that's there, and yeah, pretty busy month for stuff going on here, I guess, with potential with a guest coming up, with Kim's appearance coming up. I'm going to finalize things with him this Saturday, and finally a tour of the bug room. Save it for next week. Paraplaneta missionary. Save it for next week. And with a tour of the bug room. And Christmas is coming up, and New Year's, and all this stuff. Junior wants me to show bug. Do I even have a good bug to show right now? Yes, I have to show some bug. Quite wanting for bug right now. We could do a quick check and see if the Leucolapisma babies are on the underside of the wood in here. That's a bug that counts. I am very excited about this. I'm very excited to have added a +2 to my thysanurine breeding colonies because they have just been notoriously difficult over the years. Oh yeah, there's some babies crawling around. We'll see how well they show up. You can maybe see. Put it in front of my face. Can maybe see some babies. Yeah, you saw one crawl off the side there. Here's the adult. Hope that wasn't a baby that fell off. Here's the adult here. Again, at a glance, looks pretty identical to just standard fire brats, but these are, again, Leucolapisma arenarium, which is from the most deserty part of the desert Southwest, and actually get quite notably larger than Thermobia do. So a very exciting thing to have success with. I am very happy to have these going, and you can see the setup here. I put them on sand because I don't know, it made sense. Something that has sand in its name, maybe put it on sand, I guess, for the species name at least. And they were on just pure sand in the wild with no traces of anything else. So cotton balls. Cotton balls are supposedly an old cultivation trick for silverfish. Something about like, I think they like to wedge themselves into them to lay their eggs. And I don't know if there's any validity to that. I don't know. I guess, it's one of those things that I just do. One of the very few things with bugs that I do mindlessly and don't question. I just like, oh yeah, I'm setting up a silverfish. I should add some cotton balls in there. So they're very beautiful. They aren't quite as colorful as fire brats are, because fire brats have a little bit of green, a little bit of red. But these guys are, it's a nice yellow and black and gray. And they do have the maybe you guys can see when we're crawling around. They do have the very long filaments off the back of their body. They're extremely long, as you may or may not have seen. But yeah, so pretty cool thing. Happy to have collected them. Really lucked out getting at least a pair with 3 of them, although I did get reinforcements at one point, which is in a different container. So yeah, I think that'll about do it for this episode of the Roach Wiggle. Thank you everybody for joining me. Anybody who's going to the Michigan Reptile Show this Saturday in Taylor, Michigan, I will be there as pretty much always. And until next week, may your cultures ever be bountiful. And I look forward to seeing what you guys want to look forward to in the Bug Room Tour. So good night, everybody.